Hi everyone, I'm Francesca and thank you for having me here. And today I will tell you something about my PhD project. I'm a PhD student in the Helmholtz Center for Environmental Research and at the German Center for Integrative Biodiversity Research in Leipzig, in Germany, and I'm currently at the beginning of the second year. During my PhD, I aim to study the evolutionary responses of plant species to community diversity. It is well known that plant species vary in the expression of phenotypic traits in response to varying plant community diversity. But while the role of genetics is well acknowledged for providing raw material for possible adaptation and then evolution, plant population can actually also harbor a significant amount of variation at the epigenetic level, which can cause heritable variation in the phenotype without actually changing the DNA sequence. Because epigenetics is reversible, it can cause a rapid and irritable but transient response to varying environmental factors. It is still poorly understood to which extent epigenetic variation is under genetic control or induced by the environment. Within species variation may arise from phenotypic plasticity or genetic differentiation. It is most likely due to phenotypic plasticity in short-term experiment, but there is already some evidence for differential selection depending on plant community diversity in long-term experiment, suggesting that the strength of biodiversity effects increases with time. While it is appreciated that rapid evolutionary responses can occur in biodiversity experiments, it is not clear how the phenotype is related to the genetic and epigenetic variation. Therefore, the focus of the project is to study whether diverse plant communities have caused rapid evolutionary responses of plant species. The two main hypotheses I will test are the following. Response to plant community diversity are reflected in differences in trait expression and are heritable and adaptive. The effects of local plant community diversity, which we can observe as phenotypic differences in trait expression, correspond to changes at the level of DNA sequences and/or epigenetic modification. The location of the project is the YEN experiment, a long-running biodiversity experiment running since 2002. Here we investigate biodiversity ecosystem functional relationship in a typical European mesophilic grassland ecosystem and measure a range of key community characteristics, species traits, and processes in hundreds of research plots that can be studied across many years. It is located in the floodplain of the Saale River in Vienna, in central Germany. It consists in a pool of 60 plant species represented by 4 functional groups: tall herbs, small herbs, legumes, and grasses, which were initially sowed in different diversity and functional groups combination across the plots, ranging from monoculture level to 60 species level. The diversity level in the plots is maintained by weeding 3 times per year. Now I will present you a package of my PhD project in which we will try to quantify evolutionary changes at the genetic, epigenetic, and phenotypic level. In particular, we want to know if these evolutionary changes are in response to plant diversity and which is the relationship of phenotypic differences to genetic or epigenetic variants. For this, we chose Taraxacum officinale as a model species. Taraxacum is an apomictic species, so it means that it is suitable for exploring the role of epigenetics. An apomictic species can reproduce via sexual mode and an asexual mode. When it reproduces via sexual mode, the individuals are mainly diploids and self-incompatible and are usually insect-pollinated. And during the asexual mode, the individuals are mainly triploids, but sometimes tetraploids, and the reproduction is an apomixis with diplosphoric gametophytic. These individuals are found in purely clonal population or in mixed population with some gene flow. Some work has already been done using taraxacum officinale from the yen experiment. In particular, the individuals were coming from after 5 years of selection, and the individuals actually different in the expression of size-related phenotypic traits after 5 years of selection. The individuals, we also saw that the increasing plants diversity favored the selection of clones with characteristic of key strategy. But these studies were lacking in checking how the phenotypic differences related to the responses at the molecular level. And this is where my study comes. The distribution of Taraxacum officinale in the yen experiment is in 16 plots from a 2 species regional level to 60 species regional level. My project consists in a field experiment where we collected seed family from 16 yen experiment plots that are in these different diversity levels, so 2, 4, 8, 16, and 60. And we measured traits that were related to resource acquisition, growth, and reproduction. In particular, these traits are germination rate, initial size, number of leaf rosette, leaf greenness, maximum leaf length, and number of leaves. The project also consists in a common garden experiment where we focus on the effects of diversity and also time. We collected some seed from 2002 and from 2019 and sowed them into a common garden. The setting of the experiment is this greenhouse at the EMPSEB research station in Bad Laustadt in Germany. The greenhouse has ambient temperature and light condition and a roof that gets closed when the weather conditions are bad. The plants are grown in a common garden under shade and light treatment, which means a 40%, the shade treatment consists in a 40% reduction of light with a green shading cloth. And we measure the performance-related traits, expecting some greater phenotypic response to shading from plants that are selected in high diversity community that should be already adapted to the shadow. We will then grow a second generation from this individual from both the treatment, expecting a stronger genetic epigenetic relationship under standardized conditions to better understand the role and control of genetics on epigenetics. We will then perform molecular analysis using genetic and epigenetic STIP markers and microsatellite markers. We want to assess the level of ploidy of the population that is related to the sexuality and possible differentiation of microspecies. We will also focus on the genetic and epigenetic variation within and among the year 2002 and 2019, and how is the relationship between genotype and phenotype. We already started doing some microsatellite analysis using individuals from plots in the yen experiment collected in 2018, individuals from the initial seed stock from 2002, and also 3 sites in the surrounding of the yen experiment, always collected in 2018, to check if there is a genetic flow outside and inside the yen experiment population. We established a microsatellite marker system with 8 SSR loci, and then we start the sequences with the genetic analyzer. We have already some preliminary results that we were trying to answer to this question. Is the level of genotypic diversity affected by the surrounding level of biodiversity? For now, the answer seems not, because as you can see, it seems that there is not a real pattern. So the next steps are to study if there is an effect of the proportion of the presence or absence of some functional groups in the yen experiment, and if there is also an effect of the location of the blocks and the plots in the yen experiment, how close they are, and if this distance has an effect. We will also then check for a possible difference in trait expression within the genotype. So if a genotype can give some different phenotype. And so we will then already again focus on this relationship. Overall, we hope to The answer to our question will bring to a better understanding of how biodiversity can actually influence evolutionary processes. And now this could explain the strengthening of biodiversity itself on ecosystem functioning over time. Thank you for your attention.